All right, how we all doing today? Good.
Pretty good.
Perfect. Perfect. That's good to hear. Um, you all get hit by power outages yesterday? Yeah. No, Kate didn't. I did. Other people did. Yeah. It was garbage. I couldn't get any coffee. I was very fussy about that. Yeah. All
That's
right.
always the worst.
It is all.
Coffee.
Yeah. Although, although I do study trauma, so if not getting coffee is the worst thing that happens to me in a day, I suppose I should probably count myself lucky. So it gets worse than that. But all right. Before we get started, jump into stuff. Any questions about anything that we've covered up to this point, where we're going, anything along those lines? Okay. So just a couple of quick things to keep in mind. One, we're going to have Samantha, are you on board yet?
Yes, I am here.
You want to tell people about SI?
Sure. Yeah, we will be having an SI session tonight at 6:30. I have actually quite a bit planned for tonight, so I think it'll be super beneficial if everybody wants, as many people can. If you guys want to come, it'll be great to prepare for your exam on Wednesday. And yeah, it's tonight at 6:30. So if you have any questions, you can feel free to shoot me an email.
Okay, cool. So we've got SI tonight. I'll do a review session tomorrow. Same format. If you have questions, come through, link in. I'll stay online for however long you all have questions. We'll go through, work through that if you can't make it. So that'll start at 5. If you've got a conflict, you can't make it, we'll go through and record. If you want me to cover anything, if you're not able to make it and you want me to cover something, let me know. I'll make sure we get that addressed and then take things from there. And then quiz 2 will be Wednesday. Okay. So make sure that you've read the chapters. Make sure that you've taken notes. Make sure that you've watched all the video. Remember, we had that one day where Zoom kept kicking me off. So, all that is on, that was just a recorded lecture, so that'll be online. Make sure you watch that. Let me know if you have any questions. I'm here to help. Samantha's here to help. Natalie's here to help. Just make sure to let us know, okay? Because we want you to be ready to go for the second quiz. Format will be the same. It'll be 40 questions, multiple choice. It'll start at 11. You'll have the full class time to take it, and then we'll be done with that, okay? All right, questions. All right, people are okay. Good, you guys are capable individuals. You got it covered. All right, cool. So last time we had finished up with talking about the ins and outs of vision. Okay, and so remember what we have with our vision system. We got a binocular system. What's nice about having a binocular vision system? What does this do for us?
Allows for depth perception.
Yeah, it helps us with depth perception. If I just had sort of one eye looking at stuff like that, my ability to tease out depth starts to become compromised, right? So we've got our two eyes sort of placed on the side of our head. And so most of the stuff that happens here in the center of our field of vision is being processed by both sides of our retinas on both eyes and going to both sides of the brain, right? But if we see out here on my right visual field, and we did this is the stuff that's coming through. If I pull my finger in just right to where I can barely catch it, that's the border of my right visual field. Bring my finger in right to where I can start to just identify it if I'm looking straight ahead. That's my left visual field. What's interesting about that is anything in my right visual field is being processed on the left side of both of my retinas and going to my left hemisphere. Anything on my left visual field is being processed by the right side of each one of my retinas and is going to the right my right hemisphere. And so what we can do is, for most of the stuff that we see, is being processed by both sides, both hemispheres of our brain. But if we go through and start to present things in our left or right visual field, that information is going uniquely to one side of our brain, right? And we know because we talked about we have some lateralization stuff with our hemispheres, okay? What's our left hemisphere good at? Anybody remember?
Detecting faces. Logic.
Okay, so some logic, some detection and things along those lines, right? Really what left is good at, or one of the things that left is good at, is going to be language and sort of processing, stuff going on here. So our language highly centralized in our left hemisphere. Okay, right hemisphere, on the other hand, tends to be have some tendencies towards synthesis, organization, sort of putting things together. Okay, so what happens in some individuals, when we talked about this, your right and your left hemisphere are connected by a bundle of nerves known as your corpus callosum, and that corpus callosum is right here where we've got this contralateral functioning where stuff over here is coming from this side is going to the right, this is coming to the left. This is what allows your two hemispheres to talk to one another. But if we set up a situation, right, and as we see in this video, or in the video that I'm going to have you watch, if we set up an experiment where we have you focusing on one point in the screen, we'll call this a fixation point, okay? You're focusing right here. And then I start to present images in your right visual field and your left visual field, and your right periphery and your left periphery. What happens is that information gets sent to the opposite hemisphere, right? So if I show you something in your right periphery, if you're staring at this fixation point, I show you something in your right periphery, that's being solely processed in your left hemisphere. If I show you something that comes into your left periphery, that's only going into the right hemisphere. And if your corpus callosum is cut, as in these split brain procedures, what ends up happening is we start to see some interesting stuff going on. Now, did anybody watch that? Now, I just sent that to you not too long ago, but did anybody go through and watch a video on the split brain procedure?
Yes.
Who? Oh, Jordan? Did you say yes?
Julia.
Julia. Okay, Julia, there you go. I'm trying to see whose mic's on and off. Julia, tell me what happened in that video.
He could, I think it was his right side of his brain that did work, and then he was able to write with his left hand.
Uh-huh.
He couldn't read the word on the left side of the screen, but he can draw a picture like this a pan.
Yeah.
He could draw it and couldn't say it.
Yeah. Super, super interesting. So this is a gentleman who had surgery because of seizure disorder. And both hemispheres of his brain worked, right? But what it was is we cut that corpus callosum to try and prevent seizure activity from moving if it started on one hemisphere from moving across, right? And for the most part, this guy is fine. You would think you cut the ability for right and left hemisphere to talk to one another, you'd be pretty screwed. But in fact, he's doing all right. But what ends up happening, if I'm looking at this fixation point, and I don't have a corpus callosum, and something pops into my right periphery, Julia, what hemisphere is processing that information?
I'm sorry, can you repeat that last part?
Yeah. So I'm looking straight ahead, and something pops into my right periphery. What hemisphere of my brain is recording or processing information if it's popping into my right visual field?
Your left side of the brain.
The left side of the brain, right? And what is my left side of the brain pretty good at? Language, right? So if something, and you'll see in this video, if we present something in the right periphery of this guy, I can identify, I can say the words, I can say, I saw X, Y, or Z, all right? But if I present something in my left periphery, Julia, what hemisphere of my brain is processing the information in my left periphery?
I don't know.
So in the left periphery, what hemisphere is being processed? Where's that going?
If you're using your left side of your brain, you're using your right side.
Right. So if I present something in my left hemisphere or in my left periphery, that's being processed on the right side of my brain. Is my right hemisphere very good at language? No, it's not, right? And so what we see is if we present something in the right periphery, that information is going uniquely to my left hemisphere. My left hemisphere has language, so I can tell you what I saw in that. But if you present a word in my left periphery, And that's going over to my right hemisphere. My right hemisphere is not so good at language. And so if you ask me to say the word that I saw, I can't say it. I was like, I don't know. I didn't see anything. My right brain didn't, my right hemisphere didn't process that. But if you ask me to draw it with my left hand, I can draw the thing that popped into my left periphery. I just can't say it, right? And Julia, one more question, then I'll quick pick it on you because you were awesome and jumped in. Why can I draw, if it's going into my right hemisphere, if that information, the thing in my left periphery went into my right hemisphere, why can I draw it with my left hand and not my right hand?
Because it's the opposite. So your right side of your brain can put things together, therefore you're using your left hand.
Yeah.
Your right hand only can do it can write, but it can only process language pretty much.
Yeah, perfect, right? So if I only present information in my left periphery and my two hemispheres can't talk to one another, what I saw in that left periphery is solely contained in my right hemisphere. My left hemisphere doesn't know what's going on with that, but it's in my right hemisphere. And so can I write it with my right hand? Nope, because my right hand's controlled by my left hemisphere. But if that information is coming in my left periphery, this goes to the right side of my brain, I can draw what I saw. I can't name it. If you ask me what, and you'll see this, it's super interesting. You say, what did you see? It's like, I don't know. They say, close your eyes and draw something with your left hand, drawing a picture of whatever that was, right? Because it's in my right side of my brain. My right side of the brain is very limited in terms of speech, but I can go through and draw it, right? And so what's interesting in this video is when we start going through and presenting things in both visual fields, I'll say what I can say, verbalize what I saw in the right periphery. But if you ask me to draw it, I'm going to draw what I saw in my left periphery, right? So super interesting, a really, really interesting way to go through how so your brain is going through and crisscrossing and stuff like that, and showing how even if we go through and cut that corpus callosum, which seems like it would be a pretty big deal, and it is a pretty big deal, but our body and sort of our system is still working pretty good unless we start to play with stuff on the periphery, and then sort of things get a little bit squirrely. Okay, so go through, watch the video. It's super cool, super interesting, and then let me know if you have any questions about that. Okay.
Is that why, people who are left-handed, do they tend to make more doodles when they or something?
Yeah, that's it. Sage, is that you?
Yeah.
Okay, yeah. Yeah, doodle stuff, or sort of that right and left-handedness stuff is interesting, right? There's some research, that you'll say, oh, people who are left-handed are more creative. Because if I'm left-handed, what hemisphere is controlling sort of my sort of writing and stuff like that? The right, right? And we know that our right hemisphere sort of does a little bit better. It looks like there's some specialization in terms of synthesis and organizing information and sort of connecting the dots and things along those lines. Whereas my left hemisphere is more analytical, sort of language-based type of thing, right? So there's some interesting stuff on right-handedness and left-handedness. Your question was, do people who are left-handed doodle more often than people who are right-handed? I'm not sure specifically about that, but I do know that people have made claims that sort of in terms of creativity, of like, oh, I'm a left brain person or a right brain person, sort of speaks into some of that stuff.
Obviously, this isn't based in fact.
Yeah.
But, I'm left-handed, and I have a friend who's right-handed, and we doodle about the same amount.
Yeah.
Doodle, so
yeah.
I don't know. Sometimes we'll share a page doodling, and we'll about split the page about 50-50.
Mm-hmm. And so this, I think, Colin, this is a good thing to keep in mind about sort of any type of psychological research, right? It's easy to say, well, there's this finding, and you say, well, that's not true for me, or sort of I know people and things. Not that I'm saying this is what you're doing at all, but what's important to remember is these studies is based on big group research, right? And so just like you're saying, Colin, there's a lot of individual difference, right? People will say, that the important thing to think about, and this is important as we can move forward, if we say, hey, here's a thing, and it's pretty well established that sort of this is something or that. So like when we get into intelligence, right? What we know are certain demographics systematically perform in a different way than other demographics, right? That's not to say that everybody to a person, like this is a universal law. What this is generally doing, and the same thing with right and left hemisphere stuff, in general, it looks like there looks to be across people a tendency for this versus that, but it's always important to remember that that big group level research doesn't always sort of ring true specifically to individual people, right? And so just in terms of as we're going through and processing stuff as we move forward, an important thing to keep in mind with that stuff. Yeah. Sage, does that kind of get at sort of question some of the stuff that you were talking about?
Yes, thank you.
Cool. All right. Last stuff with vision. So as information comes through your eyes, right, and goes through and being processed, that stimuli, that stuff is being processed on your occipital lobes back here, the back lobes of your cortex, right? But like most other things in your brain, we've got a pretty sophisticated system. Not everything is being processed all in the same place. Okay, so when we're thinking about visual analysis, your primary visual cortex is back here on each one of your occipital lobes. You got a right occipital lobe, you got a left occipital lobe. And your primary visual cortex is doing some just very quick and dirty sort of basic processing. So looking at detection of edges and outlines. So if I'm looking at, say, this diagram right here, sort of me being able to identify the edges of this right here, a lot of that's going in my primary visual cortex. Primary visual cortex, sensitive to movement. This is kind of your T-Rex thing. So like if things start to move, I can sort of detect that something's moved. And some preliminary processing of color. Again, just some quick and dirty, rough processing that's going on there in your visual cortex. Okay. But then what happens is once that information goes through and gets processed quickly within your occipital lobes, what that information does is it gets passed in a couple of different streams. One we call your dorsal stream that goes up to your parietal lobe. Dorsal fin, it's up, so it goes up into your parietal lobe, and then we've got another stream of information going down simultaneously down into your temporal lobe. Okay. Now your dorsal stream, this is information. So once it's processed in your occipital lobe sends information up into your parietal, and this dorsal stream is processing where objects are in your environment and whether or not they're moving. So some additional sort of more fine-grained processing going up in your parietal lobe, looking at eye movement, visual attention, and stuff like that. Any of you who are out there who play video games? Anybody play big on video games, sort of spend time sort of doing some of that as a hobby? Okay, Jordan gave me a thumbs up, I can see. Okay, if you're doing this, like a lot of that's hand-eye coordination, movement, detecting things along those, a lot of that visual processing is going on up here in your parietal lobe. Processing and identifying sort of motion, depth, spatial location, hand-eye coordination, stuff like that. A lot of that processing, that fine-grained processing, is going on up here in your parietal lobes. Okay. You've also got a dorsal stream. So simultaneously, your occipital is sending information, doing some rough processing, and then sending stuff up and sending stuff down. The ventral stream, this is going down into your temporal lobes. And what this is going on is processing what an object is and some more fine-grained color processing stuff. So we've got regions in our temporal lobe that are very specific and look very specialized in terms of identifying specific objects, forms, and faces. Okay, so right now I see Julia's right here in my screen. Okay, Julia, I'm looking at you, right? And in this little thing, I see, when I look at your little thing there, I see your face, and then something that looks like your bed back there that you've got bunked, and some other things, right? Now, if I had to, every time I looked at an object, same thing, I'm looking at Collins, same thing too, right? And I can start to see this stuff. If every time I looked at stuff and had to say, okay, well, there's a vertical line, and there's a vertical line, and a couple horizontal lines, and there's kind of an oval thing going on, that wouldn't be an efficient system. It takes too much time, right? So we have regions in our temporal lobe that are very, very good at identifying sort of facial recognition, saying, this is a face, that's what this is, or this is an object, this is a location, things along those lines. Identifies it very quickly, so we don't have to do a whole, like, start from the beginning, sort of work through the whole thing, okay? So a lot of object identification, facial recognition. We see color perception, right? Some fine-grained stuff like that. Color consistency, right? Color consistency is if I go through and, okay, another sort of session of what's on Clap's desk. All right, so we've got this stapler right here, right? What color is this stapler? Red, right? But what if we put it in like a dark shadow or something like that? It changes, like the actual color looks like it changes a little bit, right? But we still identify that as red, right? That's stuff that's going on in our temporal lobes that's helping us identify things, some location and scene and things. So the stuff that's going on in our temporal lobes, different than the stuff that's going on in our parietal, right? And so again, what this is, it's a complex but an efficient system where we've got different parts of the brain going through and processing different aspects of this sort of very, very fine-tuned visual system that we have. So it's complicated, but it's also sort of probably a more adaptive system than just saying like, oh, everything happens there in the occipital lobe, because then if I get punched in the back of the head and it ruins my occipital, then I'm sort of out of luck, right? So the complexity of the system helps with some fine-grained nuance and things like that. Also has some adaptive functioning there. Okay. So questions about the vision stuff here? Anything that we've gone through to this point? Feeling all right with this? Cool. All right. Let's go through, click out of that.
Actually, one quick question.
Yeah.
Are the parietal lobe and temporal lobes individual cortex or no?
One more time, Colin, what's that?
Are the parietal lobe and temporal lobes individual cortex?
No. So they're different lobes. Okay, so you've got your cortex, right? Your cortex is kind of like the mushroom cap, sort of that we had talked about there, right? And you got two hemispheres. You got your right hemisphere, your left hemisphere, and then you have your cerebral cortex that runs over the top. Okay, and then you've got different lobes, right? Sort of your frontal lobe, your parietal lobe, and those are sort of demarcated by some of those deep fissures, right? And then within each one of those lobes, you have what they call different cortices. So you have your prefrontal cortex right here in your primary auditory cortex and your temporals, and somatosensory cortex and your motor cortex up here. And so cortex is used in this term, identifies just a region of that individual lobe that is sort of involved in some sort of process and things like that. That's a good, does that help kind of tease that apart? Okay, cool. Yeah, it's confusing because we're talking about this cortex, your visual cortex, but then we talk about the whole thing as your cerebral cortex, which is kind of a different thing. So when we're talking about, again, on that diagram, those different areas, those different cortices, those are just regions within on your cortex. Cool. All right. Going to switch over and quickly move through some non-visual senses here. First non-visual sense we'll talk about is audition. And so with each one of these, I want to first talk about the stimulus that's being picked up, right? So these are physical things in our environment that are being picked up by these different systems and transmitted into electrical signals that move through our neurons that we can then process and experience as sort of different sort of phenomena, stimuli, things like that, okay? If we're talking about audition, our sense of hearing, stimulus here are sound waves, okay? And your sound waves are a mechanical wave that's moving through any type of media that can be stretched and expanded, okay? Now, when we're generally talking, what's the compressible media that sound waves are moving through? Yeah, just air, right? Right now, I'm talking and sort of my mic is picking up changes in the air that are created by sort of my vocalizations, right? But if we're underwater, then it's water, right? If I shout at you underwater, water doesn't carry sound sort of quite the same, but it's just sort of differences in that waveform in the water, okay? And what we have is, in terms of studying sound audition, decibels is our standard unit of auditory intensity, okay? We're talking about decibels, so this is going to be, if we talk about sort of the power of some sort of auditory stimuli, we're generally talking about that in terms of decibels. So I can sort of, if I'm talking like this or talking like this, it increases the decibel level that's coming through. Now, it's important to recognize that decibels, this is a nonlinear measure, okay? So the difference between 0 decibels and 10 decibels and 10 decibels and 20 decibels isn't the same. 20 decibels is 100 times more intense than 10 decibels, right? So it sort of goes up, right? So just to be aware, if you're looking at, like, oh, this is only a couple decibel increase, if it's a super high sound, that's still a lot of power, okay? So decibels is our unit of intensity. But if we go through and we take a look at the actual sound waveform here, what we can do, what we know is that, and again, this is sort of the area of psychophysics, we know that pitch is based on the frequency of this waveform, okay? What does pitch determine? Or what do we experience as pitch? Like, what is pitch?
It's it's like how high a voice is or how low a voice is.
Yeah, any noise, right? So, Colin, what's the pitch of my voice?
You have a lower pitched voice.
Lower pitched voice. If I talk in a high falsetto like this, right, that I've increased the pitch of my voice. And what that's based on is the frequency or the hertz of that sound wave, okay? Low sounds or stimuli with a low pitch have a very slow frequency, right? And so it's sort of big waveform going like this. As soon as I sort of gave my high falsetto voice, that sound wave started moving much faster, okay? And so this is the frequency. This is number of cycles per second. Here we have a slow amp or a lower sound, right? Because we've got a slower waveform. Here we've got something that's higher pitch because it's moving much more quickly, okay? So we've got pitch determined by frequency, and then volume determined by amplitude, okay? Amplitude is how big my waves go, right? So if I'm talking like this, I've got waveforms going like this. If I start shouting really loud, that increases the amplitude of those waves, right? And so pitch and volume are independent of one another, right? I can have very loud, high pitch noises and very loud, low pitch noises, right? So sort of working that together. Now, our system is only able to sort of detect things in a certain range of pitch. If something's too low, we don't perceive it or sort of can't identify it. Anything that falls below about 64 hertz is too low for us to identify. We don't pick it up. Same thing, anything above about 23,000 hertz, that's too high. This is dog whistle stuff that I can't hear because it's too high of a pitch. Okay, so this is what we're looking at in terms of the mechanics of audition. Anatomy of the ear is basic third grade stuff, but just as a reminder, your pinna, that's our ears here. Some of us have bigger pinna than other pinna. But this is sort of our ears. It's going through and capturing sound from out there and directing it into the ear canal. Vibrations of these sound waves sort of transmit here, hit our eardrum, vibrates that eardrum, which then activates and vibrates our anvil, stirrup, hammer, anvil, and stirrup, our tiny bones in our ear, which then goes through and vibrates the ovular window of our cochlea, and then we get into some processing going on here. Okay. Now, when we take a look at this cochlea, right? So here's the cochlea, right? We know the cochlea looks kind of like this nautilus thing. It's all kind of rolled up just right inside our skull right here. We get information, or we get these compressible waves. They hit our eardrum, right? Shake our small bones in our ear, and then they go through and vibrate the oval window. Now, here's our cochlea. If we were to, cochlea is all rolled up, right? But if we were to unroll it, it was kind of roll out, kind of like a sleeping bag, okay? And inside of that sleeping bag is another sleeping bag that goes through, and this is going to be our basilar membrane, okay? And if we took a cross section of your cochlea, it would look something like this, okay? What we have when we open up the cochlea ends up being kind of like a tube, okay? And then you've got fluid-filled sort of spaces within your cochlea above and below, okay? And what happens is when we get a noise come in, and it vibrates my eardrum, and my eardrum vibrates the small bones of my ear, and then those small bones vibrate the oval window here. What ends up happening is the fluid in my cochlea starts to move. It causes waves in this, okay? And what we see is that inside our cochlea, and again, this is all rolled up, we have on the inside of our basilar membrane, we've got some structures here that have little tiny hair cell receptors, these little cilia, okay? Or the little receptors with these little cilia, these little filaments that go through and attach to the top of our basilar membrane, okay? So you've got this upper part is kind of rigid and solid, but the bottom part is kind of loose, okay? So when we get vibrations come in through the oval window and it starts to shake up the fluid in our cochlea, what it does is it moves the bottom of our basilar membrane, okay? And that causes these little cilia that are attached to the top of the basilar membrane to kind of move around, right? And as these receptor cells start to move around, because the fluid in the cochlea is being disrupted, it starts to bend these little cilia, these little filaments that are attached, and that's what starts to be registered as sound. That movement gets triggered and down the axons of the auditory nerve off into your system. Okay, so again, this is a really cool example of psychophysics in that everything that's sort of being translated as electrical signals and that I'm hearing right now, my own voice, is a product of some sort of physical wave being transformed into an electrical signal that then goes into my brain that I can go through and sort of process. Okay. People see kind of how the mechanics of your cochlea are working here. Okay, cool. So what's interesting, the other thing too about our auditory system, again, we don't have a good auditory system as lots of other animals, but it's still okay, right? But some complexities here. So if we go through and sort of are trying to qualify what's going on, there's a couple of different things that are activating different characteristics of sound. So the volume, okay? The volume of my voice, whether or not I'm talking like this or whether I'm talking like this, right? This is determined by the number of activated hair cells, okay? Cade, you're right here in my line of sight, so I'll ask you, why does this make sense? Why would I expect a loud sound to activate more of these hair cells than a quiet sound?
The louder is, the more movement there is within this membrane, which moves more hairs.
Exactly. Remember that loudness, sort of the volume of a sound, is determined by the amplitude of these waves, right? And if I fire through a giant wave that's doing this, right, it's going to cause huge disruptions along my basilar membrane, and it's going to activate a whole lot more of these than if I'm whispering, right? If I'm whispering, that's a very small wave that's kind of coming through. It's not going to activate as much. So we get volume determined by the number of hair cells that are being activated, okay? Pitch is coded by a couple of different things. One, the area of the basilar membrane that's activated, okay? So what carries, someone who's a physics major knows something about sound, right? What carries farther through media and media is sort of stuff. Low frequency waves, slow waves, or high waves? Low, yeah. Who shouted low at me?
I did.
Okay, yeah, Jacob. Perfect. Yeah, so if I've got a low wave, right? Very slow wave, a long wave, a slow wave radio sort of carries for a long time, right? And so in terms of pitch, if I've got a low frequency waveform, Jacob, is that going to correspond to a low pitched sound or a high pitched sound?
If it's a low frequency,
Yeah. Yeah.
Low pitch.
Low pitch, right? And so what we're going to see is the region of the basilar membrane, if things are getting activated down here at the end of the cochlea, at the end of the basilar membrane, so again, all the way down in this, that's going to be processing stuff down here at the end. That's going to be more involved in processing low volume sounds, because that low volume is able to go through and it just has more distance and it can carry up all the way down to the end, okay? Jacob, I'm going to stick with you. What about high frequency sounds? Do high frequency sounds carry very far?
Not as far as low frequency.
Not as far as low frequency, right? And so we're going to see higher frequency being processed further towards the base of the basilar membrane, because those high frequency sounds, those high pitched sounds, they can't make it all the way down there. They peter out before they get there, right? So the area of the basilar membrane that's activated, and then two, the frequency at which the hair cells fire, okay? So if my hair cells are kind of moving kind of slow, that's going to be registered as a low pitch sound. If they're moving very fast, consistent with a high frequency wave, it's going to be coded as higher, okay? So again, volume being coded by the number of cells activated, but pitch duly coded by sort of what area of the basilar membrane is being most activated, and then the frequency at which those cells are firing. So it's a complex system, but it kind of makes sense if you understand the mechanisms and sort of how sort of waves work and things like that. Any questions on this? Okay, cool. So all of this sound is being registered by your auditory receptors and these little cilia that are wiggling around. This goes down through, connects to the axons of your auditory nerve, goes through your thalamus. Remember, thalamus is a relay system, hits your cortices, contralateral functioning. In general, stuff coming into my left ear is being processed primarily in my right hemisphere. Anything going in my right ear, mostly being processed by my left hemisphere. We get sort of sounds coming in through both, so it's generally being processed by both, but some contralateral functioning here going in to auditory cortices in your temporal lobe. Okay, you don't have to worry about this, but basically, decibel levels of different sounds, and the loudest sound in the world would kill you on the spot. Like basically, if a sperm whale sort of blew off echolocation sort of right now, you would explode. That would just destroy you. So some interesting things like Krakatoa from 100, like sort of back when that historic Krakatoa eruption, 100 miles away. The decibel level of that was still something that would probably not do good things to your hearing, right? What we see with decibel levels, normal conversation about 60 to 65 decibels. If we hit 90 to 95 decibels, this is like jackhammer, train whistle stuff. This is where sustained exposure to this type of stuff can start to create some hearing loss. 125 decibels. This is going to be where pain starts to kick in. 140 decibels, even brief exposure to these types of things can start to result in permanent hearing loss. So gunshots and say a jet took off right next to you, that would be not great for your hearing, right? So as we start to get into things up here, right, testing chamber for satellites, sort of volcano explosions, rockets, right, it's not great. So stay away from rockets and sperm whales because it's bad for your hearing. Okay. All right. Going to switch it up. Move over to olfaction. Okay, this is our sense of smell. Sense of smell, pretty straightforward here. This is just a chemical sense. Okay, the stimulus for your sense of smell, these are going to be odorant molecules. Okay, so these are just chemical molecules or just chemical compounds that are floating around in the air. What happens is we breathe them up, they come in, and these odorant molecules start to connect with our different olfactory receptors, within sort of lining in our nasal passage. Okay, what we've got is our olfactory system has about 350 unique receptors. Okay, so you've got about 350 different types of olfactory receptors within your nasal passage. Okay, and what we get is different combinations of different odorant molecules that start to connect in different combinations, start to create different scents. Okay, so the type of chemicals that are coming through, it's sort of a different smell with a different thing. Think about your favorite perfume, cologne, deodorant, something like that, right? It has a certain smell, right? Now you don't have a specific receptor in your nose for your Car Noir or something. I don't know what you guys like to Michael Jordan cologne. There's not a specific receptor for that stuff, right? But what those compounds are made up of is a combination of different chemical receptors that go through, connect into those olfactory receptors, and those combinations start to create different smells. It's kind of like baking a cake, right? I have this ingredient, that ingredient, that ingredient. I throw it all together, and I get a certain thing. If I start to change things around, it changes the smell, okay? So we see our olfactory system here processed in your olfactory bulb, okay? And this is part of the limbic system. These are a couple little things that are pointing off the front of your limbic system here. And we know that our olfactory system has a close relationship with memory and emotion centers. And think about this, right? Like, again, if you've had, so your first sort of person you ever dated, boyfriend, girlfriend, and maybe that person had a certain smell. They sort of used certain perfume, cologne, or something like that, right? Maybe this was, 5-6 years ago where you dated this person. But if you're in and you like smell that specific or particular smell, boom, immediately memories and stuff like that and emotions like that start to sort of come back in, right? Same thing with, let's say your grandma used to bake cookies, right? And that was a thing, sort of grandma sort of baked cookies. And maybe now when you go and you smell sort of that same thing, boom, immediately hits in with some of those emotions, right? Makes sense sort of in proximity, your olfactory bulb, this system tightly pulled into your limbic system. And remember, your limbic system involved in memory, emotion, sort of basic life functioning and things along those lines. Okay.
So does olfactory bypass most of the limbic system, or does it have to go through the limbic system, or is it part of it?
It's your olfactory bulbs are just part of the limbic system. We didn't talk about that. It just and all the pathways go through. So it's not like things necessarily need to run through your amygdala and your hippocampus in order to get to stuff, right? Remember, the thalamus is kind of your relay station, so it just kind of takes things off through there. Other questions about olfaction? All right. Cool. Gustation. This is your sense of taste. Okay. Again, this is a chemical sense, and our stimulus are different molecules that will bind in with our taste receptors. Okay. Now, everybody thinks like, oh, my taste buds. If I stick out my tongue, oh, you can see my taste buds. Incorrect. Okay, your taste buds are not. If I go if we were all in class, I would have you sort of stick out your tongue and look at your person sitting next to you, and then people get grossed out. But if you look at the sort of my tongue, right, you see those protuberances, kind of the ridges on the top of your tongue. These are your papillae. Okay, these are sort of the rough surface of your tongue, and your taste buds are actually down in between those papillae, right? So if you go down through, so you see like surface of the tongue here, it's sort of gross and rough and things like that. If we go down and sneak in between sort of these sort of protuberances, down here is where you see your taste buds, okay? Taste buds actually have about 20 to 50 individual taste receptors. And kind of go down through down here, and this is where we go through. And when these chemicals that are registered by your taste buds start to pull down here, this is a taste bud. Again, about 20 to 50 individual receptors with little pores hanging out on the end. This is what's picking up taste and then sending information back in so that we can start to process that information. Okay, so if I go through and I taste and I sort of put something in my mouth and I taste it, like, oh, I get this taste. But it's actually got to make its way down into sort of these little crevices. What helps us those taste chemicals to get down into those crevices?
Saliva.
Yeah, exactly. Your saliva. If you do this, right, this is fun. It's gross. But if you dry out your tongue, okay? Dry out your tongue. Can you taste anything if you dry out your tongue? No, it just feels like big and weird. And you're like, right? That's going to go down through. And if I dry my tongue out, if my mouth's all dry, I can't taste anything. Okay? So that saliva is what's important for getting down into that taste, into these taste buds. And individual taste buds, we've got about 5 different taste buds or sort of receptors registering sweet, sour, bitterness, salty, and umami. This is that meaty flavor. Okay, like if you just if you have a burger or a steak or something like that, that kind of like savory type of sensation that taste, that's going to be sort of these meaty, protein-rich foods. That's your umami. That's your umami signal or stuff there. Okay. All right. Now, so taste is sort of going on and getting caught up in your receptors here. Flavor, though, this is a combination of olfaction and gustation. Okay, your experience of flavors is based not only on what you're picking up in your mouth, it's also your sense of smell. So, like, think about like if you have a head cold and you're just all stopped up, right? Do you taste things as well? No, right? It kind of puts a headache or puts a damper on stuff. Or let's say, like you have to eat something that you don't like, right? People are like, oh, I got to eat this, right? Plug your nose, sort of, this is going to go down my gullet, but I don't have to smell it, right? I got like sardines or something like that, like, sort of go through, right? And so noting that our sense of flavors, we typically smell or typically experience it, just isn't our sense of taste, but also smell coming together. Have kind of this culinary experience of sort of eating foods or drinking wines or sort of beers or sort of all this whiskeys or sort of, I like all my examples are food and alcohol, but other things too, flowers and stuff like that, right? Don't eat flowers, that's a different thing. But it's a combination of things coming together to enhance your experience, okay? And so for your sense of taste, pathways going to your gustatory cortex at the base of your frontal lobe, right? So your frontal lobe's being involved in some taste stuff like that. Okay. All right. Last, cutaneous sense. This is my sense of touch. Okay. What we see is the physical stimulus, or the stimulus for a sense of touch is going to be physical chemical temperature changes on the skin. Okay. And there's a lot of different things we can pick up in terms of our cutaneous sense. Our sense of pressure is based on mechanical deformations of the skin. So here's my arm. If I go through and squeeze my arm, right, I can feel that as pressure because there are sensors in my skin that are identifying this as a deformation of the skin. Something's changed, and we start to identify that, or our brain processes that as sort of pressure. Vibration, this is what gives us our sense of texture. So my coffee cup here is smooth, right? And so if I run my fingers across that, I'm not getting any I perceive this as smooth because my fingers aren't picking up vibration, right? If I run my hand across my beard here, right, that's causing vibrations, and I recognize this as being rough, right, because I'm getting greater vibrations in the skin. So this vibration, this gives us our sense of texture. Again, think about if you were to run your hand over a smooth countertop or a sort of brick wall. The brick wall is going to give you much more vibrations, and you'll identify that as being rough. Heat, we have a number of different heat sensors and different receptors identifying different ranges of temperature. You've got sensors that identify moderate warmth, so anything that over is about 77 degrees Fahrenheit. You've got different receptors that start to trigger in at over 88. You've got heat at over 109, things are starting to get bad, and then noxious heat over 125. So with these different temperature ranges, you've got different sensors that are specific to that. Also have different sensors for cold. Right now, this isn't super cold. This is not Wyoming cold, but you've got certain receptors that register things below 64 degrees, about things at below 82. I wouldn't call anything 81 degrees cool necessarily, but sort of different temperatures that start to sort of register here. All right, so I heard, did someone have a question?
Yeah, me.
Yeah.
What's the difference between heat and noxious heat aside from the degree?
Nothing. Just, I mean, and these are just labels that people have these are just labels that people have set up that go through and sort of to just try and differentiate between these. So, Court, it's just the sort of temperature that differentiates this. Does that help?
Yeah, and were all of these somehow scientifically constructed, or just by polling people?
Oh, the labels? Yeah, that's someone like sort of people who do sensation and perception work. That's just kind of what they identified as, like, oh, we're just going to give this sort of whatever label. So don't worry too much about sort of the individual labels on this. Take-home message here should be that knowing that you just have different receptors in your cutaneous system that start to identify sort of different ranges of temperature. Does that sound good?
Yeah, thank you.
Okay, no worries. All right. And then the last thing, you have receptors for tissue damage. Okay, this is where we start to get pain, and we got different types of pain. So intense pressure. Again, if I sort of clamped on my arm and like really sort of reefed down on it, right? Maybe I'd be like, okay, yeah, that's starting to hurt, right? So intense pressure, we've got different sensors that will go through and detect that. We've got sensors that start to hit acids and heat, sort of capsaicin. If I go through and I slam a bunch of ultra hot wings, right? I've got sensors in my mouth that start to identify that as painful. Or if I have put something on my skin that is acidic or hot, right? Stuff that picks up there. And then we've got other sensors, receptors that pick up blood, muscle damage, or sort of damage and disruption of muscles and blood and stuff like that. So I'm moving in this new place and I managed to roll an ankle, sort of the physical damage to that joint was picked up, and so the pain and the ache in that was being picked up by certain receptors that pick up damage and to sort of body stuff like that. Okay. All right. Again, different types of skin, because your cutaneous sense is picked up by your skin. Hairy skin, this is the majority of the surface area of your body, so this is just your sort of normal skin that you have. But then you got your glabrous skin, very, very sensitive. This is hairless skin on fingertips, palms, soles of your feet. Lot of much more sensitive to pressure, vibration with our hands, our feet, stuff that we use to manipulate, walk around, and things like that. Okay. All right, and then sensory pathways here through the thalamus to contralateral somatosensory cortex for this, right? Remember, your somatosensory cortex is right here on the front of your parietal. Anything I'm feeling right now, sort of the feel in the shape of this, is going into my left hemisphere. Anything I do here in my left hand going into the somatosensory cortex of my right hemisphere. We got that crossing function going on there. All right, that's it. That covers it. We went over. Thanks if you've been hanging out here for sort of as we start to go over. So lecture summary. I mean, non-visual or senses are pretty straightforward. Comprehension check stuff. Make sure to go through there. If you've have questions, hang on. Ask me whatever questions you want. If you needed to leave, take off. Sorry, so we went a little bit over, but awesome. Okay, questions.
Thank you.
Okay, good. Any questions, concerns? Yeah, Keely.
Going back to auditory.
Uh-huh.
So I was curious if, because the sound receptors go to different hemispheres,
Uh-huh.
were to have a sound in only your left ear, would it sound better than in your right ear? Because the brain has different strengths.
Yeah. So with speech, okay, speech has some pretty lateralized functioning in your left hemisphere. If we start to damage that left hemisphere, speech starts to become impacted. Sound, not so much, because most of the time, right? So I'm talking with you right now, right? And most of the stuff, same thing with vision, is coming into both ears, right? And so both hemispheres are processing sort of the quality of the sound and things like that. Now, if you covered one ear and I was just talking into one ear, right? Like say I'm talking to your right ear, that's being processed in your left hemisphere. If I shut this one, talk, it's all being processed in your other hemisphere. But remember that with the way our brain is set up, my right and my left hemispheres talk to one another, right? Because we've got the corpus callosum. And so with that system, anything that's going on in my right hemisphere, my left hemisphere knows, because they're talking back and forth. It's only when we start to cut that where we start to get that sort of some of the stuff that we see in that split brain procedure where things are a little bit tricky. But in general, for the most part, we don't see a lot of very specific lateralized stuff because our system is caught to pick up things from both sides. Now, the difference in sound that I'm getting in my right ear versus my left ear starts to help me identify location. And that starts to help. There's some processing in there that helps me say, so if I hear something over here, right, I'm like, okay, that's over here. Because most of that is coming in here and not as much as going over here. That helps signal my brain to identify location and direction and stuff like that. But in terms of audition, I don't believe that there's sort of different stuff going on where sort of my left hemisphere does something different or better than my right.
Okay.
Does that help?
Yeah, thank you.
Cool.
Is 3D, that was like in the textbook, I think it was like 3D hearing, is that stereoscopic? Or is that a different word?
I'm not sure. Does anybody know? I would Colin, I'd have to check that out. I think probably 3D hearing is like, I don't know, we all have 3D hearing unless we have hearing impairment, which is sort of a bummer. But let me check on that, because I don't want to just sort of say something to make myself sound smart, and then then it's wrong. So yeah, I'll go through and take a double check on that and let you know. Other questions? Okay, cool. So if you can, go through and try and catch up with SI session tonight. Samantha will go through, walk you through some stuff, help make sure that you're on target with some stuff with this unit. We'll have our review session on Tuesday. Again, make sure that you come to that if you're able. If you're not, send me questions that you want me to address. I'll cover those, and then you can pick it up afterwards. Other than that, Wednesday will be our quiz. Same situation as before, so just make sure that you're going through, reading your notes, have a good handle on that. And again, remember that you should be walking into that quiz feeling as though you could just take it sort of just without a notes and assistance or anything like that. Again, I think the mistake people make is like, oh, I can go through and sort of look things up in the book. Well, you can, but it's going to take time, and you're going to start get crushed on sort of getting finished. So just make sure you're prepped, and let me know if you have any questions. Okay. Sound good? Thumbs up?
Going back to the first slide.
What's that?
Would you mind pulling up the first slide again?
Is that, who am I talking? Elijah, is that you?
Yeah.
Okay. The first slide on which
That was the primary visual cortex.
Okay. Let me see here. Primary visual cortex. Okay. This one?
Yeah, thank you.
Okay. Elijah, do you have a question on this one here?
No, I just didn't get it copied down.
Oh, okay. Okay, cool. And again, Elijah, all these slides are on Wild Courses, so you can pull them out and sort of go through, take a look on that too. So, yeah, if you just go to the files in Wild Courses, you'll be able to pull all these up too. So, in case you miss anything. All right, cool. Any other questions? All right, cool. You guys have a great afternoon. Yeah, hook in with SI. Hopefully see you on Tuesday. Otherwise, good luck with quiz. All right, we'll see you.